Susan Laramore is an associate research professor in the aquaculture division at the Harbor Branch Oceanographic Institute of Florida Atlantic University. This is in Fort Pierce, Florida. Her research is focused on diseases and environmental stressors that impact marine invertebrate health and reproduction. Dr. Laramore operates the Aquatic Animal Health Laboratory located on the campus that provides industry certification to bivalve shrimp and feed producers. So with that, I will let you get started.
Okay, thank you, Danny.
I'm going to interrupt real quick because I forgot one thing. Apologies. We're going to do questions and answers at the very end. So if you guys have any questions, questions that you might have, comments, please put them in the chat box and have all of those. And we'll get to those at the end of all of our speakers. Okay, sorry about that, Susan. Go right ahead.
Okay. I'll just pretend I'm at a meeting and getting interrupted. That's okay. So I'm going to talk today about research that we did in the event of the Deepwater Horizon oil spill on early life stages of oysters and the effects on them. It's been 10 years since the Deepwater Horizon oil event, and it was notable for the amount of oil that was released, for the duration of time that oil was released, and for the shoreline that was impacted, 4 states, as you know. And all that had potential negative impacts on ecosystems and commercial fisheries because that coincided with spring spawning events in the estuaries, as well as spawning that had occurred in the open ocean and organisms that were recruiting back to the estuaries, such as shrimp. You might ask, why is a researcher on the east coast of Florida doing work on the Deepwater Horizon event? And that was because early on, it was thought that the oil would get caught up in the loop current, make its way through the Keys, and then outside to the east coast of Florida. And in anticipation of that, the state of Florida funded a number of projects, and I was lucky enough to receive funding from that initiative. We chose to use our funding to work with invertebrate larvae and with their planktonic food sources, so microalgae and zooplankton, and also the impact of sublethal exposure. Specifically, today we're going to talk about the eastern oyster and the impacts on them. Just realize this is part of a much broader project that was done. For CRASSOSTREA virginica, we looked at the impact of short-term acute exposure on fertilization, and then on development from a fertilized egg to trochophore, impact from exposure of a trochophore to development to D-stage larvae. And then we looked at the impacts of D-stage larvae and eyed larvae. And our short-term experiments were focused on D-stage larvae, and we followed them through pre-settlement, which would be the eyed stage. So just to, I'm sure most of you are familiar with the oyster life cycle, but for those that aren't, I just want to reiterate that we didn't look at all aspects of oysters and all ages. We simply looked at the planktonic phase, so right before settlement, right before they would become spat. So up to about 21 days. So I'm just going to jump right into the results and share those with you. This has all been published, so you can look at it later. Fish tank, sorry. At your leisure. We didn't see an impact when we took gametes and we exposed them to either oil or dispersed oil. We saw no impact with the oil itself, but when you add the dispersant, we did see an impact at 12.5 parts per million. I should probably familiarize you with the horizontal scale where you see WAF, that stands for water accommodated fraction, and that just stands for the oil. So the process by which we made oil and mixed it with water. And then CWAF stands for dispersed oil. That's chemically enhanced water accommodated fractions. So when you see those, that's what that means. When we exposed fertilized eggs to these contaminants and followed them through to the trochophore stage, we found that they were affected by both the dispersed oil and the oil itself. And then we took a look at exposing a trochophore and following it to D stage. And again, we saw impacts with both the oil and the dispersed oil. And you can see at what levels they were affected. We looked at the D stage larvae that did survive and looked at abnormal development. And the picture at the top is a normal D stage larvae, and the picture at the bottom is one that has some abnormal development going on. Things we looked for was convex hinge, indented shell margins, protruded mantle. This is common for any type of contaminant exposure. These are the things that you would look for. And here we also found an effect of both the oil and the dispersed oil, but interestingly enough, it was at the exact same concentration, whereas previously you had saw effects that were with dispersed oil much lower concentrations than you saw with the oil itself, if you saw an effect with the oil. This might look a little complicated, but I just want you to take a look at the moment at the first two columns. So what we did here is we exposed D-stage larvae and eyed larvae to oil and dispersed oil. And if you take a look at the first column, you can see that we did not see any effects at all with eyed larvae, even after exposure to a high concentration for 4 days or 96 hours. In contrast with D-stage larvae, we saw an effect that with more mortality occurring the longer that they were exposed. If you look at the fourth column where you see the nominal CWAF, that's the dispersed oil, you see a different picture. So you see that both stages of larvae were affected at about the same concentrations. Now, like I said, this was a busy slide, so you may ask, what is a PAH and a TPH? TPH stands for total petroleum hydrocarbons, and that's just all the contaminants that are in oil. And then PAH stands for polyaromatic hydrocarbons. That's a toxic part of the oil. And so anytime you talk about nominal fractions, which basically is just how you make the solution up, you also need to evaluate the amount of, especially PAHs, because that's a toxic component, every time you make it up, because it can differ every time you make a batch up, or if you're using oil from, let's say, the Deepwater Horizon event versus oil from a tanker spill. It can have a different PAH component, so it's important to always add that, and that's why that's there. There can be other considerations besides just mortality. And that goes back to the first couple of slides where we mentioned inhibitory concentration. So an LC50 is the amount of 50% of the population at what dose they were affected that caused mortality for 50% population. An inhibitory concentration is a behavioral activity, burrowing, swimming, feeding, anything like that that was affected 50% of the population. And that's often overlooked, and the focus is just on mortality. We chose swimming for all the different invertebrates that we looked at because, one, that's kind of an easy one to do, and two, it affects everything else down the line. It affects the ability to feed and perhaps to escape predation. Larvae are just little eating machines. That's all they do is eat and grow. And so if they can't get food, that has an impact on them. And what we found is that behavioral effects occur at 1/4 to 1/2 of the concentration that mortality itself actually occurred. If you take a look, for instance, at the WAF graph, and you see that little star, at 48 hours, the lethal concentration was 550 milligrams per liter, but you see an effect on swimming ability at 200 milligrams per liter, for instance. And I just starred the one. I didn't star anything else that was statistically significant. With the sea wasps, we had, we think, an issue with a mix-up of the 12.5 and the 25 milligram per liter solutions. But even so, the LC50 at that point was 50 milligrams per liter. So again, you're looking at a quarter to a half of the concentration elicited problems with swimming ability. So now we're going to switch gears and we're going to discuss the results of the short-term sublethal exposure experiments. This was just with dispersed oil and this was just with D-stage larvae. We saw a delayed effect in survival around day 8, and that continued on. However, it wasn't statistically significant. In the other experiments, we reared things in beakers and watch glasses and things like that for the little larvae. Here we actually tried to raise them like they would be raised in a hatchery. And so we didn't have as much replication. So statistically, we didn't see any differences, but we didn't see a trend towards problems with survival after what most people would have done with a typical acute experiments, which are 24 to 96 hours. We didn't, however, see any difference in growth. So if the exposed oysters survived, growth was the same as with the control group. So you can see the graph at the bottom that looks at shell length. And then another way to look at it is just stages of shell development. And again, you see some fluctuation, but when it comes down to the end of it, day 17, right before they're getting ready to settle, they're in the same stages. So no difference there. So in conclusion, with this set of experiments with oil, dispersed oil, and oyster larvae, we determined that dispersed oil is more toxic than oil, and that's a conclusion that most researchers have reached at this point. And that has to do with the increased bioavailability of the toxic PAHs. It's kind of like dispersant has the same effect as taking detergent and squirting it into a pan of oil. It disperses it. It doesn't get rid of it, but it makes the droplets smaller. They fall down into the water column and they don't reach the shoreline. But that has an effect for making them more bioavailable to smaller organisms. We saw effects that were life stage dependent, and you can think about the effects with the oil with the D stage versus eyed larvae. So younger stages were more negatively impacted, and we saw that with all the different types of invertebrate larvae that we looked at. If they were at an eating stage, the younger ones were more negatively impacted. We did see a delayed response with short-term sublethal exposure, but it wasn't statistically significant, so you can kind of take that either way. And behavioral impacts were seen at lower than we saw mortality. So perhaps that's something that should be considered as well when you're looking at ways to deal with oil and dispersants. Keep in mind that that wasn't the only thing that was used. There's a number of ways to remediate oil, and dispersant was just one component of that. So what were the impacts on the ecosystem and on fisheries? Well, initially, as you would expect, there were negative impacts. Oyster harvest was low. NOAA estimates that due to the oil spill, there was a 4 to 8 billion impact of oysters lost to the marketplace. You couple that with the perceived health effects for, I don't know, a couple of years that Gulf oysters weren't good to eat compared to oysters on the East Coast. And then you had a 1-2 punch with freshwater diversions used to protect shorelines, but that low salinity had the effect of also killing oyster reefs in some areas. The good news, we're 10 years out, and oysters are a very resilient species, especially intertidal oysters. They're exposed to lots of fluctuation in the environment, and they have the ability to close up for short periods of times and avoid stuff. Even though peak spawns occurred in the spring, there's also a secondary spawn that takes place in the fall, and then oysters do what we call sputtering spawning year round. So there are always some release of gametes into the estuaries. And adult oysters have been shown to clear the toxic polyaromatic carbohydrates very quickly. And so there was really no health impact in eating adult oysters if they had not been smothered and unduly exposed pretty quickly. Do we still see problems? Sure, there were some areas that haven't recovered. Oysters were smothered. Maybe the areas where oysters recruited from were negatively impacted due to cleanup efforts or the oil itself. Danny mentioned the BP money, and a lot of the states have used that to assist with recovery. Some areas might never recover. It took over 30 years for the oyster fishery to recover after the Ixtoc oil spill. So it's very dependent on where it occurred. If you'd like more information, we did have 3 publications that resulted out of this influx of money for looking at the effect of invertebrates. We published on the oysters, which I've already discussed. We also published on the effects of microalgae and on the shrimp. And that is it.
Wonderful. Thank you very much.